Okay, so this is just a quick video on mutations and how they affect our DNA. Just fill in the blanks as we go through the notes. Mutations are caused by a change in the genetic material, or mutations are just a change in genetic material. They're caused by mistakes made when the DNA is being copied. So during interphase, when the DNA replicates, it replicates something incorrectly. So in the first example, we have substitutions, and you need to write down this whole part right here. When you only change one letter is an example of a certain type of substitution. It is sometimes called point substitution or mutations, and that's just when one letter changes. Sometimes a whole segment can change, like this picture shows. This whole 3 segment switches out. So one letter can change, and that's point mutation or substitution, or a whole section can change, and that's also considered substitution. We're going to refer to it as substitution, and it's just one letter though. Alright, so you're going to do our example here and circle on this strand right here what is the change. So we can see that on this strand, the A was switched out. Everything else lines up. It's the A that doesn't belong. Alright, so go ahead and make sure you update that on your page. You'll have to just get the circle shape insert tool and just circle it that way if you don't have the draw tool. Alright, next is insertions, and an insertion is just when an additional letter is added to the sequence. So in this instance, you can see the T and the A, they still line up. The A still lines up with A, C still lines up with C. Oops, right here is there. This T should be a T, but instead a C got mixed up and is there instead, and that shifts everything else to the right, because you can see the T is still there. The T that's supposed to be here is right here, and so it's still there, it's just one spot over, so that makes everything else shift to the right. Alright, so oops, I circled too fast here on this one. Go ahead and circle where the insertion is on this strand. You can see, hold up. You can see that the U is extra right here, and that you can tell that because then it goes GUU here. There's the GUU there. So this is the extra added in letter. So here everything shifts to the right again. Alright, next is deletion. So if insertion was to add a letter, what do you think deletion stands for? So it's when one letter is removed. So looking at our sequence, T lines up with T, A lines up with A lines up with A, C lines up with C. And it should be a T lining up with a T, but look what happened. This T got deleted, and so now everything's going to shift to the left and mess up all the remaining letter sequencing. Alright, so on this strand, I want you to circle where the deletion took place. So A lines up with A lines up with A, U lines up with U, G lines up with G, C lines up with C, A lines up with A, C lines up with C. Here's our error. We removed a U in the strand below, and that shifts everything over to the left. And so now this sequence will be one letter short. There should be another letter here, and there's not. Alright, so that's deletion. shifts everything over to the left, it's going to be one letter short. All right, so harmful mutations. Mutations that cause dramatic change in protein structures are when the mutation is actually harmful. What happens is that makes defective proteins and genetic disorders, and we'll talk about them more in depth later. My question to you, are all mutations harmful? Question mark. Are they? Do you think so? The answer is no. Some mutations allow for even more variation in species and organisms, and we'll talk about that later. Kind of helps with adaptations. And then sometimes it may produce proteins with new or altered activities that can be useful to the organisms, so it's not going to be harmful. Mutations can be classified in 3 terms or 3 ways: as beneficial, neutral, or harmful. And we'll talk about those more as we talk about each type of mutation. But beneficial, neutral, and harmful are the way they can be classified. All right, bring your questions to class.